என்ன பிரச்சனை? ஐயோ ஆரம்பிக்க போறேன், தயவு செய்து கத்திராத. என்னத்த காட்டணும்?
லேப்டாப் ஸ்கிரீன் முன்னாடி வச்சுக்கோ. இதெல்லாம் சரிப்பட்டு லாக் பண்றேன். அது யார் யார் வராது?
So, good afternoon, everybody. Is it my voice is audible to everyone? Can you raise your hands? Okay, shall we start our class today? So, I'm Meenakshi, today's tutor for the class. So, I want the section to be more interactive. So, everybody will be enjoying today. So, let's go to the class now. So, today our class is about polymerase chain reaction. Can anybody know what is meant by polymerase chain reaction? Okay, so I will let you know what is meant by polymerase chain reaction. So polymerase chain reaction is nothing but amplifying a certain specific gene of interest. Otherwise, it is known as DNA photocopier. So Carol Marles is 1985 who discovered the polymerase chain reaction. So what is meant by polymerase and chain reaction? Polymerase is nothing but a DNA polymerase, which it involves a chain reaction. So polymerase chain reaction is nothing but a DNA photocopier, or which can able to produce multiple copies of DNA of the certain specific gene, which you are going to amplify it. So next is what is meant by DNA amplification in the crime scene. We can find a sample of DNA which was found in a crime scene, and it was like available in a small amount, so it could not be identified. So we can able to make it more available in the particular sequence. So we should attain more of it. So we need a PCR amplification. So certain DNA can be amplified in a certain period of time. For this, we need a DNA extraction for this process. So a certain part of gene can be sequenced in that. So how scientists solve these problems? The scientists solve these problems. What are the tissues which we can able to amplify the in PCR? Like we can able to identify the PCR is about like saliva, blood, and tissue, and hair, and even nails. Okay, how the amplification part take parts in PCR? How it can be amplified? A certain segment of DNA can be amplified by polymerase chain reaction. See this diagram. In polymerase chain reaction, what are the things which can able to identify a piece of DNA? So there are three processes which involves in a polymerase chain reactions, or DNA denaturation, annealing, and elongation. And the main process which involves in PCR is like primers, stacked polymerase, and other nucleotides, as well as MgCls. Amplifying of certain segments of DNA can be done with two methods, like polymerase chain reaction as well as cloning. So now we are going to take part alone the polymerase chain reaction. Now we are going to focus only. Now we are going to focus only on the polymerase chain reaction, which is nowadays, which is helpful in finding the coronavirus also, which taking part in the in research field also. So polymerase chain reaction, which amplifies a certain segments of DNA, which involves what's meant by amplification is nothing but numerous copies of a certain specific segment of the DNA at the targeted sequence or in short time. So DNA replication in cells has a laboratory versions, like in which as there are two parts, like it is in vitro and in vivo in the living cell. How the application will be? Does anybody know about this? Can you just tell me about it? Okay, let's go to amplification of specific target sequence. PCR, like PCR does not copy all the DNA of the sample. It just copy only the certain segment in particular DNA sequence. It copies only the few specific sequence of the genetic code from the template DNA, which is always targeted to the PCR primers. It does not require knowledge of some DNA sequence information. It always flanks from the fragment of the DNA and it to be amplified at the target DNA. The sequence of the DNA. The DNA, the certain targeted DNA has two type of primer is being used. One is forward primer and one is reverse primer. Forward primer and reverse primer bind to the specific DNA sequence and it can be amplified further. There are two organonucleotide primer, which is chemically synthesized and which is complementary to the stretch of the DNA. From C 3' to 5' primer. This is a reverse primer.
Hello.
And 5' through 5'
Hello.
to 3' is.
So how they are preparing the specific primers?
There are certain conditions it will be preparing, so we will be seeing it in the next So, two synthetic oligonucleotide primers may be chemically which is synthesized and which is already complementary to the structure of DNA. Side of 3' that is for reverse primer and 5' to 3' is a forward primer. One nucleotide of each two DNA set can be added with the help of DNA polymerase. By using DNA polymerase, we can able to add the nucleotide only to the existing that is 3' primer OH group. Why do we need two primers? That is forward primer
Sister,
and reverse primer.
It doesn't matter now whether it is RNA primer or DNA primer.
Yeah, it is that. It doesn't matter actually. We can also synthesize both the DNA or RNA, or we can able to synthesize for the nucleotide. It will be useful for the both RNA also. We can use RNA also for the reverse primer as well as forward primer.
Okay, sis. But I do have a doubt when we are using RNA as a primer, we have uracil in that. So how is that possible to get a complete DNA?
So we have to first we have to find what tissue you are having.
Okay.
Then we have to further move on the process
Okay, sis.
so that we can able to find the particular sequence of that particular DNA. So one called forward primer and the second one called reverse primer, which have the same sequence of the forward DNA, and which reverse primer which has the same sequence of reverse DNA. So which is complemented to the forward straight. So next is component of PCR. So what are the components which are involved in the PCR? It is otherwise known as thermal circular. So the essential components of PCR is PCR tube, mixed tag polymerase, and DNA sample, and primers and nucleotide. DNA sample can be collected from tissue or like blood sample, saliva, nail, and hair. We have to extract the DNA from the certain process that's involved. We can obtain DNA sample and primer, nucleotide, and tag polymerase and mixed buffer. First, we need a PCR tube which has DNA polymerase, and adding a primers which will involve in the forward and reverse procedure and process, and a nucleotide which helps the elongation of the total DNA, which able to have total two straight DNA thing, and tag polymerase which boosts the reaction of the primers and the nucleotide, and MgCl2 which involves in the mixture of
Sis,
buffers.
TAC stands for thermus something aqueous. What is that?
Thermus
Thermo
aquaticus.
aquaticus. Okay, hyperthermophile bacterium, right?
Yeah, it only obtained from the spring season, thermal spring seasons.
Okay, sis.
Next, the PCR cycle. The PCR cycle which involves, it's governed by the temperature. First, the PCR cycle which involves 3 steps, that is denaturation, annealing, and extinction. PCR proceeds only first with denaturation. When the double-stranded template of DNA is denatured by heating, typically at the 95-degree, which with the help of the 95-degree, which always divides into two striates. Next, it go at the temperature of 50 to 65 degree. It is being cooled. When the temperature is being cooled, it started to annealine with the help of primers, and it is with the help of primers when the tag polymer is attached to the particular sequence. Next, extension process. After annealing, it goes to the extension process. When the reaction is heated at a particular temperature, that is 72 degrees Celsius, by the thermostable DNA polymerase, it extends at the 72 degrees Celsius, and it will start to extend from the particular targeted sequence. So first we are thinking about the denaturation. The double-stranded template DNA denatures by heating 95 at the 95 degrees Celsius to separate at the double-stranded DNA. That is why, which breaks the hydrogen bonding. See, the region of targeted targeted DNA is to be amplified. With this two big area is to be the specific sequence. That is the region of targeted DNA to be amplified at the temperature of 94 to 97 degrees Celsius. Excuse me. Next step is to annealing. The reaction is rapidly cooled to the primer annealing temperature with the help of 50 to 65 degrees, which allows the primer to hybrid to the single strand DNA, which the primer is annealing. See, the particular sequence which I am locating is from forward and reverse primer. The primer will always align only to the particular sequence that is complementary to that of the DNA template at the degree of 50 to 65 degree. And the extension, next process is extension. The reaction is C2 to the temperature. It is only report upon the DNA polymerase which is used. So it takes place only at the 72 degrees Celsius with the help of this enzyme. At the step, the DNA polymerase synthesis a new DNA strand complementary to the DNA template. That is, which is very, very complementary to at the temperature of 72 degrees Celsius. At the end of the PCR reaction, the specific sequence will be accumulated in the copies. In only 20 cycles, PCR can produce the 2 power 20 copies of the targeted million things. When when the DNA template can be split into 2 DNA template, and which is split into 4 and 4, 8, and 16, it goes on the amplification exponential. So performing a PCR steps involves identification of the location of the targeted sequence in the particular DNA template, and primer design and primer specificity, and a PCR optimization. Post PCR and analyze the result using agrostep. After PCR, we have to find the specific disease. So we can use agrostep electrophoresis and PCR troubleshooting, and start your PCR and visualize the result by age. For example, you want to study a mutation in DLG3 gene.
Meenakshi, hi. Sorry to disturb you.
No, ma'am.
The participants are asking for 5 minutes break. Can we resume again at 6:30?
Okay.
So everybody please join the same manner at 6:30. The participants are asking for 5 minutes break. So let's take 5 minutes break and rejoin at 6:30.
Okay, ma'am.
Okay, thank you. नीचे ना वैसे देखते हैं होगी एक्सटेंड So I am ending a meeting for a while. So let's join after 5 minutes.
Okay, now is everyone is audible? Can you hear my voice?
I can hear you.
Okay. Okay, let's start the first process in PCR, that is denaturation. The particular in DNA, the particular sequence has been done. In the particular sequence, the particular targeted sequence at the temperature of 95 degree, it is get elongated. Next, the process called aniline. Aniline, which at the temperature of 58 degrees Celsius, it is being cooled down, and the primers is attached to the particular targeted sequence. That is 5' to 3'. It is a reverse primer has been attached. To 3' to 5', the 5 forward primer has been attached. So next process is extension. With the help of tag DNA polymerase, it gets elongated from 5' to 3'. It gets elongated. In the forward primer, it gets elongated to 5' to 3'. At the targeted sequence. Next, the extension at the 72 degrees Celsius, it's been elongated as much as possible to the side of a particular sequence. When it returns to 5' to 3', the reverse primer which is added to the targeted sequence, it's also elongated to 5' to 3'. And with it is also elongated at the forward primer, that 5' to 3' at the 5' range. As this process, like denaturation and extension and alanine, which involves again the lagging, not in one cycle. It involves many cycles that each DNA can split into 2, 4, 6, 8, 16, and so on. And this process of second cycle, which always involves, there are totally 40 cycles, minimum 40 cycles it can be done at the 48 hours. How will you make sure that your targeted sequence is amplified? And it is very important to know your product size and why. Our targeted sequence size should be 350 base pair. for example, we are using targeted sequence with the help of certain aggressive electrophores with the help of
So
markers.
it is mandatory that we should have only 350 base pairs.
Yeah, it's mandatory.
Okay, if we need those base pairs which are less than 350 base pairs, then what we
Actually,
have to do?
actually, the thing is that now we are using only 350 base pairs. We have to analyze our product size. With the help of the product size, we have to prepare primers.
Okay, so we have to add primers to bring that to 350 base pairs.
Yeah,
Okay,
not to build the 350 base pairs, but
okay.
we are focusing only on the targeted sequence size. In a
Mm-hmm.
DNA sequence, the DNA sequence, the two-strand DNA can be divided into single-stranded DNA. In the single-strand DNA, we are targeting only the particular sequence target. Okay. In that
Yeah.
particular sequence standard, it should be like I'm taking a sample. Okay. In that sample, in the sample, the sequence size is 350. Okay, the targeted sequence size is 350. It depends upon the sample that which we are attaining, the DNA sample.
Okay, sis.
So PCR is a method which is used in molecular biology, which able to do many aspects in research as well as in many research areas of identifying a particular microorganisms, particular genes, then etc. Like for example, like if I want to identify a DNA, if I want to identify a specific genes or like a specific DNA sequence, what are the steps which involves in that is first we have to extract DNA sample and we have to directly go for the PCR method. PCR method is a precision method which is you make some multiple copies of DNA of the particular sequence or a desired sequence. Then the most easier advantages are it is easier methodology, but it takes 48 hours to get a desired sequence product. But it is also very simple and it's very sensitive. And the validity it takes much more time to operate the procedures. And we have to use the reagent and equipment very precisionly. And the PCR applications. So PCR not only helps in identifying the microorganisms, but it also involves as genotyping. And there are different type of PCRs, like real-time PCR, like these PCRs, which is useful for many aspects in biotechnology, as in cloning, mutation detection, sequencing, and microarray forensics and panity tests. PCR optimization. There is no single condition that optimal for all PCR reactions, but it only involves what kind of lab temperature that we are going to do that process. But the temperature in which you are going to do it, it doesn't matter while doing that PCR in the lab. So the primer design guideline. So how we are going to design the primers? First, want to know about the primer sequence, that is flanked this sequence. Then we want to know what is the length of the primer. As a guy asked about the primer, what is the targeted sequence, which involves only the base pair, that is 350 base pair. But we have to find the primer length also, and GC percentage, and GC clamp, and melting temperature, and aligning temperature. So primer design guidelines. Primer sequence must be complemented to flanking sequence to over the targeted region. So complementary sequence should be always is equal to the flanking sequence of the target region, which we are going to, which the reaction going to elongate. Then complementary sequence between which we have to avoid during the primer designs are complementary designs between the primers, and there should be a misprimer, that is we repeat or run or mismatch at the third end and the cross homologue. So what is the primary length which involves? So it is generally accept that the optimal length of the primer should be 18 to 25 base pair and not too much long or short. And a primer design guidelines. What is meant by GC content? A number of GS, that is CS, the primer as the percentage of the total base. And it should be only the 40 to 60%, that is GSC percentage. Then GC clamp. GC clamp is presence of G or C bases, that is guanine and cytosine species within last 5 bases from third 3' end of the primers and not more than 2 GS or C' S. For example, see this in CGA and GA, that is 3' and which involves C and G. It presents within the last 5, which involves in the third dashed end of the primer. And the primer design guidelines. What is that? Melting temperature. What is TM? TM is like nothing but a melting temperature. The melting temperature ranges between 50 degree to 60 degrees. Generally, produce the best result actually. The maximum difference between the primer base is like 5 degrees Celsius. The melting temperature of my primer can be calculated from the following formula is G plus C into 4 and A plus T into 2. And this is the melting temperature for the particular primers. The aniline temperature. The primer melting temperature is estimated of DNA hybrid stability, which always determine only the annealing temperature. It depends only upon the length and the GC compositions of the primer. If the composition, that is annealing temperature, is too high, it will be insufficient to produce primer template of the hybrid templates, hybrid products. If the annealing temperature is very low, the non-specific product has a high number of base pairs. We can able to attain the product right here. It is a specific product which is operated at a high annealing temperature. The low annealing temperature, we can able to have non-specific product with a high number of base mismatches. Now, you should be able to answer the following questions. How the application will be done? You can answer it. You can answer it in chat box.
I will decide again. Anyway.
And how you will determine your target sequence, and how the application is specific for the certain segments, and what are the requirements to carry the PCR. You will be get to know about these things from the above PPT. And it is a small homework that I want to give it to you. Suppose you're performing PCR that begins with one double standard of the DNA following DNA, and draw one cycle of PCR reaction below the following diagram, and label the DNA and the primer and what is happening at the yeast. I want this homework to be done, and you can just mail me to this so that it would be it's like a not like a homework, it's like a game, like so that you can able to understand what's the concept of the PCR. So I would like to thank the ClassTree, who gave me this wonderful opportunity to share my knowledge in this platform. So if you have any doubt, you can ask me in the chat box. Okay, guys, thank you. I just want you to fill this thing. Okay. Okay, like I will ask, I will repeat the former questions. Okay, suppose you're performing a PCR that begins with one double standard T of the following DNA template. Okay, if there is a single DNA template, okay, the targeted region that I have mentioned here, okay, and for this thing, you have to draw one cycle of PCR reaction below the following diagram. That is this cycle one, which involves denaturation, annealing, and extension. That is cycle one, which involves temperature 95 degrees Celsius, and the second thing is annealing, which cool downs, and third thing is extension. And this process, label the template and the primer, what is happening at each step. And this was your homework. It's not like a homework, it's just like knowledge what you gain. So thank you guys, and I hope you enjoyed this class. If you have any doubt, you just mail me to this number, to this mail ID. That is Manohari Meenakshi 1904@gmail.com.
Thanks, Meenakshi. The class was very informative and we enjoyed your class.